It's gone.
Just a sec.
All right. All right. Welcome, everybody. It's my pleasure to do the introductions today. My name is Mike Dietrich. I'm chair of HBS here at Pitt. Today's ALS lecture is given by Dr. Angus, who's chair of the Department of Critical Care Medicine here at UPMC and the University of Pittsburgh. He's also Associate Vice Chancellor for Healthcare Innovation. He's a distinguished professor and holds the Mike Mitchell P. Fink Endowed Chair in Critical Care Medicine. He also has secondary appointments in medicine, health policy and management, and clinical and translational sciences, and is a senior editor at JAMA. He's a world-renowned clinician, translational, clinical, and health services researcher. His research interests include clinical, epidemiological, and translational studies of sepsis, pneumonia, and multi-system organ failure, as well as health services research in the organization and delivery of critical care services. He's a fellow of the Royal College of Physicians in the United Kingdom and received multiple national and international honors, including election to the American College of Physicians. We're very happy to have him here to speak with us today about clinical trials.
So thank you very much, Mike. So this is, I'm now in about hour 3 in my first ever exposure to a philosophy department. So I'm having lots of fun so far. When I was asked to give this talk, I was asked to put a little abstract together, and I wrote it about a couple of weeks ago. And then about 2 days ago, I thought I have to start putting some slides together. And I read the abstract, and I said, God, am I going to talk about all that in 50 minutes? So this will be a little bit of a talk where I'll try to hit on everything I said in the abstract, but there'll be a certain amount of drive-by, and feel free to ask questions afterwards. I have no financial disclosures. I do want to start off by just clarifying, does everyone know what an RCT stands for? It stands for both a randomized clinical trial and a randomized control trial, which are the same thing. And this really gets down to semantics and the fact that a bunch of linguists at JAMA, for example, felt like if you're randomizing, there has to be more than one group, so you don't need the word controlled. But the Lancet still likes to say controlled, and if you're doing it in humans, it's obviously clinical. But it's the same thing, it's an RCT, and we're going to talk about it. What I'm going to talk about is the following. I'm going to try to make an argument to you that healthcare is basically divided in two. It's divided into part of healthcare responsible for science or knowledge generation, and another part responsible for practice or knowledge translation. The RCT is held up as the gold standard for knowledge generation and serves as a fulcrum between these two parts. It's basically the final hurdle that discovery needs to cross before it's released into the world of practice. And both premises are problematic. The RCT provides poorer quality knowledge than is typically understood, and separating knowledge generation from knowledge translation is oversimplistic and flawed. Randomization remains elegant and probably vastly underused, but RCTs have to be smarter, safer, so that they're better tolerated and easier. And knowledge and generation and translation have to become integrated and iterative. I'll now try to prove it. So part 1, the mess we find ourselves in. So society's 2 broad goals for healthcare could possibly be stated as the practice of medicine, provide all care today as optimally as possible given current knowledge and resources, and the science of medicine, find better treatments for diseases to improve the length and quality of life as quickly as possible. Now, implicit in these goals is I want you, the healthcare system, to care for me and or my loved one. And I want you to do it in a way that if I show up today, care for me with the best knowledge possible. And if I show up tomorrow, try to be even better. Now that's slightly more abstract, but it comes into focus if you ask it the other way around. If I show up today, would I be happy getting yesterday's care? And the answer is no. So even though I might not have understood it, I actually do buy into the fact that I want you to keep getting better. and keep bringing better care to you. And society has some strong mental models to rely on how this all works. So TV docs have been blending the science and practice of medicine for ages. They're inspired by a William Osler-esque model. He is still held up as maybe the great clinician scientist of the 19th century, and they are driven by compassion and calling. They apply their encyclopedic knowledge and flawless judgment to ensure their patients receive the best current care. And in instances, usually once an episode, where there's a problem, faced with a disease with no good treatment, they deploy their insight and brilliance to find a breakthrough just after the final break for the ads and before the conclusion. Does everyone know who these two people are? Oh, can we move this? Can I move it up in the corner? Where should we be? Do I even need to have this thing visible?
Press the one line and it'll go away.
It'll just go away. All right, so who is the guy top left?
Marcus Welby.
Marcus Welby. So, oh, now I need to. So, it of course is a flawed model. So William Osler was practicing in the late 1800s, so the 1860s, 70s, 80s. And by the time 1900 came around, the entire number of biomedical papers being written per year was still 1,000 or less. So if he decided he wasn't going to go out too much during the week, he could probably get through 1,000 papers a year. So I could believe that he was responsible for that entire encyclopedic knowledge. By the time Marcus Welby comes along, we're already producing 100,000 papers a year. So even if you're really bored and you never go out, I don't think you're reading all 100,000 papers per year. And indeed, this crusty old Scotsman, Archie Cochrane, had kind of lost it with the biomedical profession by the 1960s. And he wrote this book commissioned by the Nuffield when he was at Oxford that was published in 1972. And it was a critique of the National Health Service, the practice of medicine. And he basically said, they have to stop making stuff up. And he said, doctors must base what they do on RCTs. Now, the British Medical Journal did a review of this book, and they wrote, the hero is the RCT and the villains are the clinicians who fail to carry out such trials or ignore the results. And you can't quite hear the voice of the person writing it. So I don't know whether they said with disgust that Archie Cochrane was calling into question these physicians as villains, or whether they were saying, yes, Archie Cochrane is totally calling them out. But this was it. This was when sort of evidence-based medicine got launched. Indeed, many of you may know that evidence-based medicine is largely called the Cochrane Collaborative, etc. It's all after this guy, Archie. Now, Archie decided that he needed to get into this in part because he felt like most of the evidence base upon which physicians were basing the practice was either pulled straight out of their head, or at best, they were relying on largely observational medicine, which of course was entirely correlative as portrayed in this cartoon, where people who are married are happier. Is it happy people get married, or marriage makes you happier? Who knows? And he said, instead, you need to really think about randomized trials, in part because of what's widely credited to be the first randomized trial in medicine, the so-called Bradford Hill trial, led by Bradford Hill. And part of that team was Archie Cochrane. This study was published in 1948, and it was based on the fact that after the Second World War, Britain had lots of open TB. And it had a new friend, the United States of America. The United States of America had invented streptomycin. And they were producing a whole bunch of it. And as is often the case in the friendship between Britain and the United States said, we'll help, but they didn't quite help enough. They sent a bunch of streptomycin over to the UK, but not enough to treat everyone. And so the British government basically said, we should do a random lottery. And the first randomized trial is essentially randomizing the supply of streptomycin they had. And so a bunch of patients got streptomycin, a bunch of patients didn't. And for open TB, if you randomize to treating versus not treating, you can imagine there's a big difference in the outcome. So this was like a pretty positive trial. The argument went as follows. The design of the trial was to take a whole bunch of patients with disease X and borrowing from Ron Fisher and from his experiments in the 20s in agriculture. If you were to toss a coin, you could divide into two groups of patients who many physicians would think would be producing two identical groups of patients. The non-identical groups of patients, in estimation, they would be identical or at least similar, but they would be the idea would be that there would be a hopeful balance, and we'll come back to this in a minute. Some would get treatment, some would get placebo, then you'd follow them up, and then you could count up how many people had recovered, how many people hadn't, and you could, in comparing the results, you could attribute the difference in results to the fact that you gave some treatment. So randomization balances in expectation the measured and unmeasured variables, and the key here is the unmeasured variables. All the confounders that you didn't even know were confounders are still going to be subject to randomization. Thus, the RCT is providing an unbiased estimate of the difference in outcome caused by the treatment. There are some large caveats, and we're going to get into them, but this is the essence of the RCT. Now, not to be outdone, just like 18 months or so later, the United States also weighed in and passed in 1974 the National Research Act. And one of their first orders of business was to create a National Commission for the Protection of Human Subjects. That's about 12 people or so who got in a room at a place called Belmont in 1976. They sat there for about 3 days. They had a few more meetings afterwards, and about 3 years later, they finally published the Belmont Report. And the Belmont Report makes for a great read, and it addressed 3 issues. The second issue was the basic ethical principles for respect for persons, beneficence, and justice. And the third issue was how to actually do it. Operational rules for informed consent, assessing subjects, etc. And it's beautifully written. And it's beautifully written even through 2023 eyes. You can still see it online. The first issue was they decided they should start the document by defining what is research and what isn't research. And so they defined all of medicine into research versus practice. And they kind of gave a big pass to practice. They sort of said practice is when you're solely enhancing the well-being of an individual with reasonable expectation of success. That was not Archie Cochrane's position. Archie Cochrane's position was that the practice of medicine was a mess. But the Belmont Report was saying, no, these docs are like, they're only motivated by good things. Okay. So that really set us on a path that almost 50 years later now, science and practice of medicine are essentially completely separate parallel universes. To people outside medicine, they look kind of similar and they might not even realize that they're different, but they're totally separate institutions. It's the NIH versus Medicare and the Centers for Medical Services. It's different people, researchers versus clinicians, clinical research coordinators versus nurses, and so on. They're governed by different policies and procedures, by different funding and so on. And this separation has really been codified throughout the years by structures like the Belmont Report that was totally ensuring that patients' interests are not usurped by unscrupulous scientists. And obviously the Belmont Report coming after Tuskegee, et cetera, that was crucially important. But it's kind of assuming that practice is perfect, certain, and aligned with patients' interests. So the good news is that a lot came from this structure. So today, we're now testing pretty much all the ideas before they come into practice with RCTs. In 2022, clinicaltrials.gov filed 38,000 brand new RCTs. We've been thinking of new questions to ask at a very high frequency. I can't even imagine what 38,000 questions are. Pretty much all FDA and drug and device approvals are now supported by robust RCT evidence. And I don't know if this is a good thing or a bad thing, but we're committing 50 billion a year to it. That's how much we spent last year. You could say that's not enough, or that's like an atrociously large number, but it's a heavily invested number. And there's been a huge gain in methodologic conduct, in commitment to ethical oversight, and in the quality and transparency of reporting. So by many metrics, the infrastructure and the superstructure for clinical research has really yielded great gains in performance over time. But even when they're well conducted, there are some huge issues with RCTs. And tied and interrelated, it's hard to separate them. There are problems with this separation of science and practice. So let's start with problems with RCTs. I'm going to talk about, it's not so much the RCT itself as the answer that the RCT gives you. And the first is, there's the Goldilocks problem. The RCTs gives you answers that are both too narrow and too broad. They also give you answers that are won through too much effort. They're too hard to do, and in part because they're really hard to do, we're not getting enough of them. You would think 38,000 sounds like a lot, but actually there's many more questions that are not being answered. So in terms of too narrow, and I won't go much beyond this, but a common criticism is that when you do an RCT, you've cherry-picked a population, and you've tried to find a population which get a very large treatment effect, and that doesn't reflect what would happen in the real world. At the same time, people complain that you're getting an average treatment effect, not an individual treatment effect. And this I'm going to unpack further, because I actually think this is often a little bit misunderstood. And if we were to unpack this, it actually helps us solve number one as well. They're too hard for researchers and funders, and that they're expensive, slow, and draining to do. You get white hair trying to do them, and part of that is because they run on an entirely separate infrastructure from clinical care. And they're too hard for patients and for participants. You don't really want to be in a trial design where if it's really going to help, you were in the wrong group. And indeed, even in cancer, where we think that there's high participation in clinical trials, this paper written by our current Surgeon General, before he was our Surgeon General, estimated less than 2% of cancer patients are in trials, and it's far less than 2% in all other diseases. And then in terms of too few, and I'm also going to come back to this in a bit too, but just to give you an example of how many clinical questions have no RCT answers. If I were to take something like treatment guidelines formed by consensus committees, arguably the most famous is the American Heart Association Get With The Guidelines, which have been updated every year for several decades now, and it's on how you take care of patients with acute myocardial infarction. It's a pretty big disease in the West. You would think there would be a pretty robust database. 11% of the 60 or so treatment guidelines are based on RCT data, and all the rest is based on expert opinion. Are you shitting me? Like decades later, after all the progress in cardiovascular care, it turns out that, pardon my French, by the way, but it turns out that a ton of our evidence for how to, a ton of our guidance for how to take care of patients is still just based on expert opinion. And of course, many populations are never studied, rare diseases are still understudied, and after initially perhaps studying too much in vulnerable populations, now we don't study enough in vulnerable populations. Okay, so I said I would unpack this question of too broad. I already alluded to cardiovascular research. There were some really amazing cardiovascular trials going on in the 80s and 90s, like the ISIS trials and the Gusto trials, and with it, a big corresponding drop in mortality from acute myocardial infarction. And people were really making big breakthroughs in targeting all sorts of diseases related to atheroma. And things seem to be going splendidly. And then this interesting little single author paper by a neurologist from Edinburgh, Peter Rothwell, cropped up in the Lancet in 1995. And he asked the question, can overall results of clinical trials be applied to all patients? And he doesn't mean all patients outside the trial. He was saying, can they be applied to all patients in the trial? And he said, like, well, what do you mean? The question was carotid endarterectomy, which was in the sphere of cardiovascular disease, or in this instance, cerebral vascular disease, looking at atheroma. And it turned out that when people were having strokes, they often had strokes because they had plaque developing first in their carotid arteries. And a bunch of industrious, hardworking neurosurgeons and vascular surgeons said, I could take that out for you. And so they developed a procedure where they could, basically, you could image the carotid, and if it showed that there was thickening on the inside with the atheroma, with a little plaque, they could go in, they could slice, they could pull the atheroma out, and hey presto, you would get an expansion of blood flow, and your risk of stroke would go back down, and you wouldn't end up having a stroke a year later. They did a large definitive trial, European trial. There was also a North American trial, also said the same thing. It said, hey, big reduction in the chance of getting subsequent strokes. It seemed great. Now, these surgeons are very confident, but they're not perfect. And sometimes when you make these little slits in the carotid, and you go and you try to pull the plaque out, sometimes like little bits would come off. And they would head northwards and you would have a stroke. So the surgery had like a known complication of stroke. And the goal of the surgery was to avoid stroke. And so when he looked at this and he said, you know, if I go on the baseline risk in the trial based on how much narrowing there already was, which was predictive of how likely you were to have a short-term stroke versus a longer-term stroke. And I divided you into someone with a less than 10% risk of having a stroke in the next year versus more than 15%. It looks like the absolute risk reduction with surgery was not a risk reduction in very low risk patients. Actually looked like you were increasing the risk of stroke by 1.4%. And that most of the gain was in the patients at very high likelihood of having a stroke, which kind of makes sense. If you were to put effective surgery on a y-axis, and there's lots of opportunity for benefit, but also opportunity for harm. And then on the x-axis, you were to plot the baseline risk of having a stroke. The average trial is sitting in a sweet spot that gives you a big green return for the so-called average treatment effect. But all those patients in the trial are on a continuous line of risk of having stroke. And it turns out that although the RCT was positive, there were many patients who were worse off despite being in the arm that was the better arm. Now, it gets worse. This would be if the patients were evenly distributed, you'd already be scared. They're not. In nearly every trial population, there's always this shift where most of the cohort are sitting to the left with a long right tail. So that, for example, the median is always sitting to the left or always lower than the mean for a likelihood of having an outcome. So this is true in trial populations because it's true in underlying disease populations, both for acute diseases and chronic diseases. Diabetes. There are people with severe, awful diabetes and people with mild diabetes. The people with severe, awful diabetes die shortly afterwards. So if ever you try to take at any moment in time a snapshot of all the people with diabetes, there's more people with mild diabetes than there is with severe diabetes. This is true almost every single disease. So for example, I work in the ICU. If I take something like the Apache risk of death score, and I take it for patients who are already sick enough that they have to be in the ICU. So they're all sick. I've already shortened this population down to my narrow cohort. Even within that cohort, I will still faithfully reproduce more patients will be mildly terribly sick than really, really terribly sick. This is always the case. And so if we were to go and look at a reanalysis, say 10 years later, of a drug approved in sepsis called Zygris from the so-called Prowess trial, it turns out when you plot the risk of mortality and then you look at treatment versus placebo, all of the benefit is accrued by just a few high-risk patients. 2/3 of the patients are over in the lower part of the curve where there's marginal benefit or harm. So what are the implications of a misleading average treatment effect? We only get an average treatment effect from a trial. That's the primary readout from the trial, the primary outcome. How much did it change on average? Well, it's a little unpleasant to consider the penalty that was being paid by the patients enrolled in the trial. And you immediately ask yourself, could the trial have been safer? But you also reflect on, this is the last time we're doing a trial. If it was a positive RCT, the FDA gives it stamp of approval and now it's released. And the label is the entry criteria for the trial. So now everyone in the population that meets those same entry criteria will get the opportunity to keep duplicating that same slightly scary looking distribution of risk within the trial. And so that makes you ask the question, could we have had smarter answers from the trial? So let's go back and try to remember, what's the problem we're trying to solve here? The problem we're trying to solve here is an individual treatment effect. We're asking the question, patient comes to see the doctor and says, I have a problem. The doctor thinks, I think I have a diagnosis, I think I have a treatment, and I'm asking the question, what's the outcome Y for person I given treatment, T equals 1, versus not given treatment, T equals 0? It's simple. It's a very simple question. How will their fate change after given the therapy? The problem is we can't observe individual treatment effects. As Carl Pearson said, everything in the universe occurs but once. There's no absolute sameness of repetition. And this is Paul Holland's fundamental problem of causal inference. The only person who has access to this is Michelle Yeoh in Everything Everywhere All at Once. She can access multiple parallel universes. So she can give her patient the drug in one universe and not the drug in the other, and then skip forward in time and see how it worked out. No one else can do it. So we end up, in theory, we want to be getting an estimate for the individual treatment effect, an EITE. That's the goal, surely. Okay, so if we go back and look at our randomized trials with our individual patients, let's say it's for a binary outcome like live or die in a sepsis trial or something. The first patient, we would like to know, would they have survived both with treatment and control? In other words, they were immune to the disease. Versus the second patient might have going to be die with control, but then if I give the drug, they get saved. So they're saved by treatment. Someone might have died no matter what, they're they're doomed. And someone was actually going to do just fine. But then I gave them the therapy and it had a fatal side effect and they were harmed. That's what, in theory, I would want to know. Those are these individual treatment effects for each individual. And of course, I don't have that. If I give treatment to one patient, by definition, they can't be in control. So therefore, they're immune or saved, etc. So I have these impossible conundrums. So how are these RCTs working again? So it's just useful to go back and sort of reconsider that sleight of hand, because at the end of the day, I'm wanting to treat individual patients, and yet I'm being guided by this RCT. So what is the RCT doing? And there's lots of people will unpack this. Angus wrote a lovely little deconstruction of the RCT a few years ago. So remember, what we're getting is the average outcomes for those treated, an estimate E of all of the outcome Y for all the people in the group that are treated T equals 1, minus the same thing, the outcomes in the absence of treatment for those who were in the control arm. Now, that same equation could be expanded out to say that the difference between the control group and the treatment group is the difference between the treatment group's outcome with treatment, which we saw, versus what outcome they would have had if we hadn't given them outcome, which we can't see, plus the difference between the two groups had none of them ever seen any treatment. Now, those two extra terms which are unobservable are actually the same term, which is what would have happened to the treatment group had they not been treated. And since they're expressed in this equation as a minus and a plus, they cancel out. And to the extent that this is a randomized trial, had it been done repeatedly, i.e. in estimation, groups, terms 3 and 4 are 0. The two arms, had they never been treated, if they're just drawn as randomly from the sample, they effectively cancel. They're drawn from the same distribution. And so thus the difference in average between the groups is the same as the difference in average between the treated group getting the treatment and the treated group not getting the treatment, the counterfactual of what would happen. That's the magic of the RCT. And it's a genius measure of average treatment effect. When people write about, and it's basically Ron Fisher's insight, like he's a complicated person, but they would generally say it was a remarkable insight. And he's basically borrowing from Gauss so that he can get it's unbiased by confounders, and it is imprecise in that it's only one estimate of the treatment effect, but it's drawn from a predictable standard error, a frequentist distribution, etc. But that's all it is. it's a genius measure of the average treatment effect. And if you do everything else right in the RCT, so there's not other confounding, you get this robust estimation of the average treatment effect. So let's go back to prowess. The prowess event rates, 31% in the control arm, 25% in the treatment arm. That's a 6% absolute risk reduction. There was also a 1% increase in fatal bleeds. Xigris thins your blood. It will work almost accidentally like an anticoagulant. So that means, let's imagine we're doing infinite sampling, and then we're expressing it per 100. There are 7 saved by Xigris, 1 killed by Xigris, 24 who are going to die no matter what, and 68 who are immune. All right. None of them were 6% less dead. Everyone was either dead or alive in one of these 4 unmeasurable groups. So I don't really want the average treatment estimate from prowess. I want some sort of conditional average treatment effect, or a CATE. And in other words, I want the average treatment effect conditional upon some pre-randomization variables, which I could have used in the trial had I known them, or at least after the trial, I could have derived from the trial, so I could use in the label and how I'm going to use it in practice. And my ideal set of covariates in my prediction model would parse 100 sepsis patients as follows. 7% the cohort with 100% chance of being saved, 1% with 100% chance of being harmed, and 92% with 100% chance of no effect. And then I wouldn't make as much money for Eli Lilly as they were hoping, but I would be able to just nicely use the drug in the 7 patients with sepsis who would have died and now would survive. I'm not going to get them, but I would still be plenty happy if you gave me the following rule. 21% of the cohort with a 33% chance of being saved and 0% chance of harm. So a prediction rule that found 7 patients who would be saved and 14 other patients who otherwise would not have benefited but not have been harmed. So if I did that for every 3 patients treated, I would save 1 life. And 79% of the cohort would have a 100% chance of no effect or harm, and I would never treat any of them. That would be great. What did we get? Well, what the FDA did is they approved the drug in the upper 37% for risk of outcome, not for risk of change in outcome from the therapy, because they didn't know that. So that group over on the right that I showed in the earlier graph with the 13% absolute risk reduction, in whom we have an unknown individual risk of harm or saved. All the fatal head bleeds could be in that group. And the remainder with the lower net odds of change, even though there was a smaller benefit, they actually banned, they excluded them from the label. Okay, so what did we say the actual job was here? I thought we were trying to get an accurate estimate for the individual treatment effect. That should be the job. Well, I'm already saying that we get an 8. We try to get a conditional average treatment effect, maybe, but we're still a long way off getting an individual treatment effect. Furthermore, the entire clinical research enterprise focuses almost exclusively on average treatment effects. The example I just gave you of the FDA restricting the label is one of the only times they've ever done that. They almost never restrict the label inside the RCT. They take pretty much carte blanche what the RCT's entry criteria were, which is, by definition, going to be the full ATE. So at the minimum, you can start designing trials that as a primary output are going to produce more than one average treatment effect. Power the trials and design them so that you're going to generate CATEs, separate 8s conditional upon select patient features, for example, severity of illness. But still remember that the distance from the 8 to the CATE is dwarfed by the distance from the CATE to the IT. Who's spanning that gap? That entire distance is therefore bridged by physician judgment. Okay, so those physicians, when we give them a trial, we're only giving them an average treatment effect. I also mentioned we often don't even give them a trial. We're not doing enough trials. So I already said by 1960, there's 100,000, you can guess what's coming next. We're now writing 1 million papers a year. No one's reading those. Medical knowledge is apparently doubling every 3 months. I'm just going to tell you, the knowledge may be doubling, but the knowledge inside the heads of doctors is not doubling every 3 months. There's a massive increase in healthcare choice and complexity. When the National Research Act was being signed in '74 and Archie Cochrane was writing his treatise on the National Health Service, ICD-9 was being launched. ICD-9 captured all possible diseases known to humankind, and there were 15,000 of them. We now use ICD-10 with 69,000 codes. When we order a laboratory test to find out which diagnosis it is, we've got over 10,000 tests to choose from. And if we're trying to pick a treatment, the FDA has 20,000 approved drugs. Now, there's a bunch of generics in there, but if I just focus on brand new individual compounds, 2,000 brand new individual compounds have been approved since I graduated from medical school. If you asked me to write them down, I would be incredibly proud if I could think of 200. I couldn't even tell you how the 200 all work. I can't even list them for you. It's massively complex. And so we're not really asking questions half the time of drug A versus placebo or drug A versus drug B, but rather we need to be thinking about which combination of pills should I take. When I admit someone to the ICU, we still don't quite know what's the best set of empiric antibiotics for the person at risk of sepsis to start them on. But that's a drop in the bucket. I am at the same time as I'm ordering the antibiotics. On the ICU admission orders, you can't even read this font size because the standard ICU admission orders has over 50 things on it. Have I done something on? Sir? Yeah, I don't want you to read this. That's my, yeah. We're making decisions around all sorts of things, and there's uncertainty about all manner of these, including how they all interact with each other. And so that brings me to this issue of separating science and practice. Clinical decision-making is inconsistent and unreliable. It is far worse than alluded to in the Belmont report. Clinicians actually are making 100 care decisions per day under uncertainty. It's remarkable, between 80 and 120. That's true whether you tour around with an ICU team rounding in the ICU, or you follow doctors sitting in outpatient cardiology clinics. Turns out our brains start to fry after making about 100 choices under uncertainty per day. And like all decisions, we're making them under uncertainty. We're victims of bounded rationality. We're limited by our human cognitive skills, our imperfect access to information, time constraints, and bias. We have all the same problems that were identified across the road here by Herbert Simon and by the burgeoning fields of decision sciences and behavioral economics. And so current healthcare practice in the United States, physicians are relying on a set of heuristic rules of thumb that are not necessarily in the patient's best interest, that are not informed by all available data, and that are not consistent. There's a long lag before new evidence is incorporated into practice, if ever, with no incentive structure to promote adoption of new evidence. And only a little over half of the time are patients receiving even current recommended care, even the care recommended based on expert opinion alone. The National Academy of Medicine thinks that we dropped $1 trillion in 2021 on unnecessary, ineffective, avoidable, or harmful, in quotation marks, care. Okay. And again, by having these two separate organizations, there's no incentive to collaborate. There's a broken incentive and collaboration structure, gross inefficiencies in the conversion of biomedical knowledge into improved health. In this paper that ran in Nature in 2008, the then director of the NIH, who preceded Francis Collins, Zerhouni, said, he quipped, yeah, it's right. The NIH isn't really responsible for health. Maybe we should just rename ourselves the National Institutes of Biomedical Research. The other thing that happens is people have labeled this bad practice of medicine as a problem of knowledge translation, or KT. The problem with that framing is that it assumes that the job of generating the knowledge is fine We're just not translating. But as we've already said, for example, an average treatment effect from an RCT puts far too much burden on the physician to then bridge the gap to an ITE. So it's actually a two-way problem. So many of these things existed before, and then came COVID-19. And COVID-19, by compressing time, by suddenly giving us a brand new disease that we didn't have any treatments for, it brought into stark relief what's actually been happening all along, that people don't sort of see it properly because of the scale. But it's very obvious to everyone that you have to treat patients as well as possible and learn, find out what therapies help. And there was a priceless White House press conference when they were talking about hydroxychloroquine, which doesn't actually work, as we'll get to in a minute. But it sounded so good, this was this anti-malaria drug, that at one point someone asked, so should we start giving hydroxychloroquine, or should we start testing it? And the then President Trump and the then advisor, Tony Fauci, basically both spoke. And one said, we should start treating patients with it. And the other said, we should start doing trials. And then they like stumbled, and then they looked at each other. And it was this priceless moment where they were caught in trying to make this potentially false choice. And that is the classic dilemma in decision-making under uncertainty, which again, Herbert Simon's colleague, James March, so nicely characterized at Carnegie Mellon as the exploration-exploitation trade-off. The elusive solution of trying to work out when you should be exploring, trying to find new things, versus exploiting the knowledge you have, just doing what you think you know already. And that is a balance or a trade-off. It's a ratio. So if you take responsibility for the ratio, you're responsible for the numerator and the denominator. Now, in optimizing decisions under uncertainty, or solving for exploration exploitation, nearly everywhere else, that's characterized as complex adaptive systems research. It shows up in organizational science, in mathematics, and evolutionary biology. It's how tree trunks explore and make decisions about how deep down they should look for water versus use ATP to just extract energy. Now, it turns up everywhere. It's all through artificial intelligence. It's the root of multi-armed bandits, Markov decision processes, etc. It's all disciplines are exploring, optimizing, learning and doing, except in healthcare, because we divided the structures. No one is actually empowered to find the optimal trade-off. That's always true in medicine. It's just particularly obvious during a pandemic. And so that's the mess we're in. So now, what are our options and our opportunities? I'm afraid the slide doesn't say how we're going to fix it. I'm not sure. But a couple of things have happened, and actually COVID gave us a little window on how we can make our way out of some of this. So we need a research instrument that is robust, practical, attractive, and relevant to practice. It generates causal inference, it provides answers across broad populations, it provides answers that project likely benefits for individual patients, or at least for more narrowly defined types of patients, allows for complex combinations of treatments, it lowers the barrier of entry to patients, and it leverages the infrastructure of, for example, usual clinical care to lower costs and logistical burdens so we could imagine doing more. And you then say, well, now that we have digital medicine, can't we just like pull all this data down from the EHR and sort of look things and basically use big data clinical decision support? And lots of people have thought, well, this could be the solution. Lots of people said this will be learning health systems. We'll just look at all the decisions we made last year and work out how we should have made our decisions. And of course, the problem here is back to the same problem at the beginning of, do you have adequately robust causal inference? And a whole other talk could have been about paths to causal inference in the absence of randomization. Suffice it to say that while there's lots of room for instruments other than randomization, there's also still huge problems with relying solely on observational data for causal inference. So if we stick with RCTs, since that's the talk, what could we do with what's going on in RCTs? And there's two flavors. One flavor was these so-called point-of-care clinical trials, where they're looking inside the electronic health record for clinical moments that alert the trial machinery, where there's a moment in time, which is actually copying the old analog cardiology trials of the 80s, where I'm clutching my chest pain to my chest and I'm admitted to the hospital and thinking, oh, admit to the coronary care unit. Now's the time to also randomize to try IV atenolol or streptokinase, etc. And the VA got into this action about 10 years ago with giving like little EHR prompts when doctors were trying to order sliding scale or insulin for poorly controlled diabetics. And they said, we see you're trying to order insulin for your diabetic. Do you know there's uncertainty around how to give insulin? Would you like to have the patient be randomized? And this has actually been taken further by Picori, and we've now actually got into a couple of big trials and thousands of patients doing this, but they're still totally old-fashioned 50-50, one-to-one RCTs. All they are cheaper for the researchers. Nothing else is better. But it's still cool that they were nested in the EHR. There's also another family of trials called these so-called adaptive platform trials that try to take master protocols so that there's almost like, when you do a trial, it's like you built a football stadium to play a single game of football. And then you dismantle it and you drink beer afterwards. You say, that was like a great game. And then a week later, someone says, you want to play another game of football? Sure, let's go build a stadium. And then we'll play the game of football. And so master protocols are, we could build the stadium and we could leave it up. And then we could play like more than one game. We could use all that same infrastructure. And there was this increasing movement of adaptations beyond just things like early stopping rules using Bayesian statistics, and maybe you could blend it all together. And those are so-called adaptive platform trials, but they were really only being used in a very narrow way in things like breast cancer in phase 2 trials. And it was focusing on efficiency with almost like sometimes tiny little sample sizes to try to work out what should we take forward into phase 3 trials. So maybe you could blend the two. And so we started envisioning a few years ago, okay, we're going to have a platform that allows for perpetual enrollment and continuous learning. It'll be adaptive in that it's going to match odds of success to odds of assignment. It'll be multifactorial in that we can test multiple treatments and subgroups. It'll be embedded both culturally with care in the clinical care environment, aligning with care and potentially leveraging the EHR, but still randomized for causal inference. And that's a remap design. So in the middle of this engine is this so-called response adaptive randomization. And it's worthwhile just talking about that for a second. So if we go back to our trial again, and we think we have equipoise, at the start of the trial, it's 50-50. If you ran a trial and it was positive, in the end, you were more than 99% sure that A was greater than B, what was going on in the middle? Now, under traditional frequentist designs, or you can't look under the hood, multiple testing, multiple loops, you have to pay an alpha penalty and so on, but let's look under the hood. So let's do a trial of 400 patients, A versus B. We're 40 patients in, and it's alive or dead, and the 41st patient is up. Who still has equipoise? Who's happy that your patient, the next patient is your uncle? You're working out, are you in your uncle's will? One thing you're not working out is, do I have equipoise? I think probably everyone in the room is thinking, I think the odds that A works are better than B. And you'd be correct. At this point, the probability that A is better than B is 78%, which won't get you published in JAMA, Lancet, or New England, but it's not 50-50. So imagine if for the next 40 patients, the trial took a bet proportional to that first 40. So it's like a weighted lottery where now we're going to have, say, 78% of the next 40 patients will get A, because if A is really working, we will load on A. And if it were really working, after 80 patients, you are done. Now you're at 99 point, you were going to be 400 patient trial, but you have totally blasted past with only 80 patients and you're done. So you stop the trial. Now there are caveats. If the second 40 was flat or in the opposite direction, then the trial continues and the next bet swings back closer to 50-50, and you can seesaw back and forth during the trial. Now, when there's two groups, we all remember when we're doing little power calculations that the sample size is driven by the smaller group. So this whole thing seems like for naught in a 2-arm trial. But who's doing 2-arm trials? We just looked at the complexity that we're faced with, all the drugs we're choosing among and all the subgroups. So this would not be helpful if we were asking questions in homogeneous cohorts with 2 arms. Who's doing that? It's very interesting if we're doing multiple arms and multiple subgroups, which is in reality what we're facing. And so response adaptive randomization, you could have a randomization rule where you're testing in 3 arms and you start out a third, a third, a third. The data accrue, they go into a pre-existing Bayesian model, then gives an updated randomization rule after each block of time. Each block of time is like being in a little Markov state. It's actually like running an MDP. And at the end, and in the first period in the MDP, you've got these 3 options out of it. And now you've accrued data, you update it. And now you jump into a new state. The new state is the second part of the trial where you've now got updated probabilities for exit. And that goes on iteratively through the trial. So the odds are weighted towards the best treatment. All sorts of things can happen. You can start de-weighting MC. You can have rules for triggering when a new arm comes in. So like a higher dose, the higher dose doesn't start unless you've already seen a monotonic relationship where the other doses were outperforming the lowest dose and so on. And you can have rules for dropping arms, and you can have entirely different weights for different strata. So in the same trial, you could be working on generating different weights for different substrates, or different strata of patients. You'd also love to embed the whole thing in the EHR, if you could, and have it all be triggered. So in simulation, if we're doing an 8-arm trial, where 2 of the arms are much better, 20% mortality, and several of them are horrible, in this little Monte Carlo simulation for a 2,000 patient trial, the black line that's flat across the line here would be the 250 patients per arm in a traditional 8-arm trial. And the gray bars are how many patients are actually put into the trial. The trial had no knowledge that arms 5 and 8 were much better, but it was taking bets during the trial based on what it was seeing. And you can see how it was massively pushing patients into 5 and 8 as it was learning. Now, at this point in the trial, the power is actually relatively similar between the 2 trials, as noticed by these red dots here. But there are 80 fewer deaths in the response adaptive randomization. So this is a much safer trial than a standard allotment. Okay, so these remap designs are potentially smarter in that they consider many different treatment options. They vary the options depending on the patient, and they're potentially safer in that they're probably playing with those, probably the winner. And if outside the trial, no one knows, and the trial is learning, the safest place to be is in the trial, because it's the only place that's taking a bet based on the accruing evidence. So this was all in theory. We had designed a trial, remap CAP, community-acquired pneumonia, that we were setting up that would run in multiple places in pneumonia. And then if a pandemic hit, we would flip into pandemic mode. We got this thing turned on, felt like weeks before COVID-19. We were not ready, but COVID hit and we flipped into pandemic mode March 1st. This trial is up, this is slightly out of date now. It's more than 380, it's something like 360 sites, but it ended up running all over the world. We coordinated it at several different places. And we were basically studying all hospitalization patients with COVID, dividing them into severe and moderate, with a primary endpoint of organ failure 3 days that was a combination of being able to survive and or avoid organ dysfunction. And we divided it, there's a little bit of nomenclature, into different domains. So one domain could be a choice of antiviral, another one could be giving steroids or not, another one could be giving fancy immunosuppressants, and you could be eligible or not eligible for multiple domains. And then within those domains, you'd be randomized to different interventions, which meant that you would be getting a regimen. It would be like choosing a starter, choosing a main course, and choosing a dessert. Only they're all brought at the same time and there's multiple different options. We could be testing those for different subgroups. So then you get these complex interventions where someone might be A1, B1, C1, A1, B1, C2, and so on. So then a remap cap journey for a patient, they show up, they're assessed for eligibility for different domains, they're randomly assigned, then we follow them through time, their data goes into a model, the model updates after 2-3 months, and now all the randomization weights change. So how have things gone? So this slide is also slightly out of date, but we've now enrolled about 13,000 patients, but here you can see there was about 11,000 patients in. And on average, people were in about 2 separate domains. That's why there's about 20,000 randomizations in 10,000 patients. Some patients were getting 5 or 6 domains, all randomized simultaneously. So in the moderate patients, we were testing vitamin C, we were testing anticoagulation, antiplatelet strategies, immunoglobulin. How many regimens are on this slide? The model also accounted for if you were ineligible. So it's vitamin C, yes, vitamin C, no, or vitamin C not in it. So there's like a dummy variable. So there would be 3 in the top one. So you don't need to do the math. The point is there's 8,600 random options in this trial. This is much more complex and much closer to the reality of the complexity that clinicians are managing. In the ICU cohort, there's almost 200,000 random options being tested across in the trial. And the trial has produced lots of results. So in JAMA, we published on the benefits of steroids. In the New England, we published on the benefits of IL-6 receptor antagonists. We came back to the New England, 2 more papers showing that anticoagulants were beneficial in the low risk, but actually harmful in the high risk. Back to JAMA with convalescent plasma not helping, antiplatelet therapy not helping, and then showing that hydroxychloroquine was actually killing people. It was actually harmful. At UPMC, where we're running this with no clinical research staff that were deployed in the hospitals. There was a little team available online for doing consent by video consent, but we were enrolling totally inside the EHR and totally with frontline staff. And so there are many hospitals on here that are in rural parts of Pennsylvania or inner city serving predominantly minoritized populations with high enrollment. So this design can now suddenly reach lots of people. And the outcome was then immediately turned into policy. When we had a positive result, it turned on the guideline and then everyone followed it afterwards. So that 17-year gap from results to practice was shortened down to essentially 1-day gap. Okay, so there's definitely issues you could ask. Can you put a whole trial in the EHR? And the answer is, actually, I understand why people get nervous about the EHR, but these are just technical problems. There's technical problems about getting CFR11 compliant, so you need to be able to take snapshots of the EHR because the EHR is constantly updating. There's definitely a lot of technical issues, but they're all manageable. And even a standard clinical case report format is largely abstracted from the EHR. And you can always increase the quality of an EHR by spiking it, by specifically asking people to do some specific extra data collection, which in fact was what we were doing in COVID, where we impressed upon UPMC that every COVID patient needed a standard intake of the CDC baseline criteria for COVID. Institutional commitment. We had huge institutional commitment during COVID, although whether there will still be institutional commitment is a very serious issue. Ethics. There's many things to say about ethics, but the narrow thing, could you get this approved and was it easy to consent? It would actually went far more easily than anyone expected, in part because responsive adaptive randomization, it turns out, is very intuitive to patients. If you say that the trial will preferentially assign as it goes forward the therapies that seem to be working best, that like, you say, don't you always do that? Statistics and design, the very fact that we've got this, we're still learning and there were lots of issues, but it was definitely doable. And it would not have been doable even as much as a decade ago. There's no doubt that even some of the software for modeling this has only been coming online since about 2010, 2011. A lot of people didn't know if we could report and disseminate the results. It's a huge ongoing trial. And yet these are individual reports with only part of the trial. Every time we were tackling things like consort flowcharts and all the standard reporting guidelines, we had to sort of break new barriers on how we report those. But we've totally gone, and not just us, but the recovery trial out of Oxford did the same thing, the active trial program from the NIH, these adaptive platform trials have absolutely broken new ground on reporting. And then funding, there's actually been quite a lot of funding, and there's even been more funding for things going on afterwards in this same model. Oversight and regulatory approval, data safety and monitoring boards definitely found that they weren't getting paid enough. They were largely voluntary and they were getting a lot of work thrown at them. And that probably needs more work. Regulatory approval, some people in regulatory authorities have still been suspicious of this, but there's definitely been movement forward in this space. And for example, the current FDA director, Rob Califf, is a huge fan of these designs. And then integration with the other clinical research programs, a lot of other clinical researchers were nervous about the appetite that these platform trials have and how much space they take up. But actually, at least in the one we've been involved in, many of the big ICU clinical trials groups have all just folded into the trial and it's gone much better than we thought. So in terms of next steps, there's 2 or 3 things. Moving REMAPs and adaptive platform trials into other diseases. All of clinical research is still a little slow after COVID in other diseases, but actually many REMAPs are taking off. I got a call last week from Harvard about something called REMAP ILD, REMAP interstitial lung disease that I'd never even heard of. And they were saying, oh, we've designed this whole new trial, and they're definitely taking off. Testing things other than drugs and devices. This whole talk has been about testing drugs. I think that's like a tiny part of the variance in healthcare delivery. We can't even decide, there's so many things we do by rote, like how often a nurse measures a patient's blood pressure. These tons of little decisions that are being made under uncertainty. In theory, all of them could be tweaked. Different prompts could be randomized and so on. Revisiting ethical approval and human subject protection. I do think that the Belmont report was really brilliant for what was seen to be inside research, but it's actually done a disservice to the entire implementation science agenda and to this whole notion, like it's created something called quality improvement that, like, I don't know what quality is and I don't know when it's improved, and there's all sorts of issues. And so it's worthwhile revisiting. There are some trial design improvements that need to be made, and then addressing superstructure and institutional incentives, including really trying to envision and create a true learning health system where you're optimizing the learning doing trade-off based on causal inference. So just a couple of examples to finish. There are these thought pieces on tweaking almost every prompt that's put in front of the EHR, and that could totally be done. There's a huge behavioral economics agenda around healthcare decision-making. Nancy Cass in particular has been thinking very thoughtfully about a new ethics framework for learning health systems, and now is actually on secondment to the FDA, and we can only cross our fingers that something will happen in that space. And then I just want to give you a little window on what we've been thinking about for advances in REMAP design. So we've now been funded to do a study called REMAP Vax that will launch in the fall on why people don't get vaccinated. It's targeting African-American populations in Allegheny County. And we're going to be simultaneously randomizing at the patient, clinic, and community level. There are actually many problems where you might want to hit multi-level and you don't know the relative effects of them. You don't know whether a patient level incentive is conditional upon whether you also do a community level intervention and so on. And so this design that's like a cluster randomized design is going to randomize alternate patient reminders while also randomizing the primary care clinics to different interventions and 10 communities in the Pittsburgh area to getting sort of free vaccine clinics set up in local churches. We still need to get closer to ITEs, and we need to get smarter about CATEs. So the Canadian Institute for Health Research has funded a big part of our REMAC family that was running the anticoagulation studies to launch something called ATTAC-CAT, which is in regular pneumonia, but again on anticoagulation. And we're in the middle of working out a Bayesian model that actually prospectively is modeling across 3 axes of heterogeneity. The source of the pneumonia, the overall severity of illness, and also the likelihood that coagulation is mechanistically driving them towards an adverse outcome. And the trial will give a readout in this cube. So it's actually, even if they're dichostomized, there will be 8 separate Kates coming out of the trial. And then we're also, this is only in simulation, but we're modeling how to minimize the altruism penalty. So this is extending on William Press's work on multi-armed bandits that was in PNAS about a decade ago. And we're comparing in simulation, if you have uncertainty around 3 or 4 drugs, in the beginning, no one knows what works. And over time, you can learn. And every day, some VIP shows up and says, I don't want to be in the trial. Just tell me what's best. Your estimate of what's best becomes increasingly precise over time. Really wealthy pharaohs still couldn't buy iPhones. But so preferential access is only so good if the knowledge doesn't yet exist. But you can then create in a simulation model, what's the best possible outcome if money were no object and you could always get the experts to give you the best possible estimate. In other words, let me look under the hood, see how the trial is going, see which looks like the winner, and then I'll just take the winner. I don't want to be randomized. And we're setting that as the best you could hope for. And then we're playing with the different response adaptive randomization rules to get as close as possible to giving you the same odds of preferential outcome as the VIP. So in conclusion, I've tried to tell you that organization of healthcare is in 2 parts, science and practice. That was well intended, but it is highly problematic. The RCT is elegant but limited. There are novel designs that retain randomization, but are, I think, potentially smarter and safer. These designs better optimize decision-making under uncertainty. And the next steps include further improvements in design, but still the need to address profound structural, ethical, and logistical challenges that exist within the status quo. And I would leave you with the goal of a true learning system, health system, would be to fuse the practice and science of medicine, thus enable continually updating, seamless, bi-directional knowledge generation and knowledge translation. And the RCT re-envisioned can remain the fulcrum for this aspirational goal. Thank you.
Questions for Dr. Angus?
So you talked about randomization, but what about blinding? And I was
Right.
wondering whether this makes something. So it seemed that maybe you were in your advanced models you were looking at groups with blinding anywhere, or at least not double blinding. So is that right? And is there a problem? And my understanding is one reason you're not allowed to look under the hood for a trial, right? Is you're going to ruin the blinding procedure. So doctors are going to discover that the drugs are really working, then they're going to have a then you're going to have bias, you're going to have to see the effect of the doctor because you know that it's doing well, and so
So these are 2 related bits. So there's a couple of parts. So the looking under the hood isn't just the issue of whether you would be blinding and using placebo, but rather just there's a penalty for looking and then trying to make a definitive conclusion based on a particular sample size, because the estimate of the error bases on pulling from the known frequency distribution for the eventual sample size, and there's a different frequency distribution. There's a different error around every other sample size. And so if you look more than once, you have to, there's an alpha spend. So you can actually look under the hood in a traditional trial, but you pay a price every time. But I use that term looking under the hood, and you're also right that if you could actually see what's going on, you could be biasing. That would be seen as a post-randomization confounding effect. I deliberately stayed away from all of that. That's the whole issue of internal reliability in an RCT, in part because I was saying, even if the RCT is done well, you still have all of these other issues. And I sometimes think that people actually get too caught up in internal threats, internal validity threats to RCTs, in part because they plagued RCTs through the 70s, 80s, and 90s But now, if anything, internal validity threats are actually relatively rare. And I sometimes think we've overcompensated. Sometimes we put in placebo where we've overthought it. That the placebo gets in the way. The advantage of placebo is it shields you and or the provider and the patient, if it's double-blinded, from accidentally then doing other things. Sometimes you actually want to understand the composite effect of an intervention, including all the other things it would be doing downstream. And so if you want to know the net effect of the intervention, not mechanistically what the intervention did, but all the ancillary things, sometimes you deliberately wouldn't want to have placebo. And then even when you do think about placebo, sometimes having placebo or not having a placebo wouldn't matter because there's nothing either the patient or the doctor could do. So some so-called hard outcomes are thought to be relatively impermeable to knowledge of assignment. And then, as you also alluded to, sometimes you just can't practically blind. But I just decided to not talk about blinding today. Not that it's not important.
Oh, okay. So I'm not sure I'm totally following your map, so I could be misunderstanding what you're recommending. But so you started out with this like a gap between knowledge production and knowledge translation, and then I feel like the solution, on one hand, I think it's super interesting and it's exciting. But am I right that part of what's happening is we're just handing off, like clinicians don't have to make decisions about how to treat patients anymore, because the trial is just going to make it for them, right? And so then I feel like there's part of, because it's like, it's not really bridging the gap between, like, it's not bridging the knowledge translation gap, which is part of your point. It's just like clinicians can outsource certain clinical decisions that they would have to make about treatment to the trial, which is going to assign the treatments. And then the other part of me is like, well, yeah, this sounds good and it works, but then if we think it's good and it works, why not just like let healthcare be a trial forever, right? Like, why wouldn't all treatments just be going into the computer super brain that can then tell us what to do, and then we're kind of like getting rid of clinicians?
Ready to say? You might have just answered the question. Yeah, so I think there's a lot to be said for what is the role of the clinician. In the same way as you could say, what is the role of the professional? What's the role of an airline pilot? So increasingly, we won't let an airline pilot fly a plane the way they used to. They're still there, they still can override, but they play a very different role from the way they did in the past. Physicians, I would argue, are being charged with a ridiculously long list of cognitively complex tasks for which they cannot possibly have the right information set. So first of all, even if we had run all the trials already and we knew all the answers, I still think it would be really clumsy to ask the physician to just remember it in their head. I mean, even if we had reduced most of the uncertainty, the recall task is probably almost certainly better done by the EHR than by the clinician. So no matter what happens, clinicians, we ought to be offloading tons of cognitive tasks from clinicians, because the healthcare that we want to provide today, I don't know anyone smart enough to be on top of it. So I totally think there should be an appropriate recalibration of what physician autonomy is. Now, that's already been going on separate from all of this. Even in my practice, there were many things that I used to do longhand early in my career that are now all automated inside the EHR. Decisions on anticoagulating patients, doing insulin control. I used to sit and longhand think through, where do I want their target to be? How much insulin will I give? Now I just say insulin sliding protocol. And all, so there's all sorts of things that are being automated already. And when all of these came in, all sorts of people said that would be a threat to physician autonomy. In fact, there are even papers on our fellows no longer learning the same way because there's been so much angst about with protocols coming in. So separate from the question of reducing uncertainty, even if we had certain knowledge, there's too much of it for physicians to retain in their head, and we need to be automating huge parts of it. But The point in this is to go one step further and actually say that there's huge amount of things for which we don't yet have adequately accurate estimates. And this notion of physicians deciding what treatment to give, I'm very nervous about that, because when you say that, it sounds like the physician is calling on something that they know. And I don't know what it is they know. Like, I don't know how a clinician can read a trial in the New England Journal of Medicine about a drug they've never used before that gives an average treatment effect, in which some patients are being harmed and some patients are being helped. And there's no part of me that thinks, thank God, the physician is going to decide which patients should get it. When I read that, I think, oh my God, we're just letting the physician who's never used the drug make a guess about whether they'll give every patient that meets the entry criteria of the drug, or they'll try to make some reasoned guess. Now, I'm not saying that there isn't wisdom of the crowd and there can't be some nuanced contribution by the physician and or the team, and I'm not looking to get rid of any of that. But I definitely think that we leave clinicians, of which I am one, alone with far too much decisional complexity and not nearly enough information.
I'm sympathetic to that. I just wanted to make sure that I see the clarity what you're suggesting. But I guess the thing that I kind of wonder about too, beyond physician autonomy here, which would have to be reshaped significantly, what about patient? I mean, there's a little bit, I guess one way of the worry that arises for me is you're like, well, they can't make this decision with all of this uncertainty, so let's clarify the uncertainty. But and the science is giving us more certainty. But like, I don't know, in our profession, we worry a lot about the science always giving us uncertainty, because sometimes it seems certain for a long time and they get everything right and then it's wrong, right? And so autonomy exists in part, I think, in frameworks where we recognize uncertainty, like its existence is part of what allows for autonomy, because we don't think there's always a clear answer. And so then I just wonder, like, do you see what these remaps are doing, or these computers, when they're making recommendations, is they're like removing enough uncertainty so no longer do physicians have to make these kinds of decisions, so they don't need as much autonomy? And is that also going to be true for patients? Because the uncertainty's just been removed. So like, why would they need to have a say either? Because there's just the best treatment.
Right. So there's a lot in what you said there. So a couple of things. So first of all, even if, so you don't, there's lots of ways of thinking about these designs. So if you're in a space where you're not testing experimental therapies and you're testing among existing therapies, first of all, before the trial launches, if we just stick with physician autonomy for a moment, there's all sorts of things you can do with physician autonomy to make them feel involved. So for example, you could decide that you're going to run within a big group practice. You're actually going to sit with them and have them discuss among themselves. What are the decisions that they take, which they kind of admit on reflection are based on a little bit of a hunch, and they know that their colleagues do it a different way. And they might recognize that there are 2 or 3 things that they do that they've never been able to decide among themselves. And one of them quite likes giving drug A, the others give drug B, drug C, but they don't think the guy that gives drug A is crazy. And they admit that one of them could be better than the other. And then you say to them, well, how would you feel if we ran it where we were going to give you a randomized prompt to give A, B, or C? And when we suggest that you give A, even though you normally give C, we would say, why don't you give A this time? And furthermore, you can override it. And then you would have a randomization of the recommendation, which is a little bit like some cluster RCTs where you don't necessarily always get total uptake, but you can still get causal, as long as you don't have crushingly high refusal rates and differential refusal rates. There are ways to do designs where you can, first of all, seek input from all the physicians about how they feel in the first place, how much they feel they want to engage in answering the question. And then you can even have a design that still gives them like an override button, which is a bit like flying the plane and still being able to take back control. For patients, it's quite, it's quite interesting that some of these more novel adaptive platform trials actually were taking hold, first of all, in the hands of patients. So where they first got up and running was through the Susan G. Komen Fund with breast cancer. And it was actually patient advocacy that was driving this. And many other patient foundations like the Multiple Myeloma Foundation have actually been embracing this. And some of them think that there's more autonomy in setting an agenda for research than actually being at the mercy of a physician who is less transparent with them. And some of them have even felt like for some rare diseases that this is a way of getting our diseases fixed by generating more evidence base. I'm not trying to, I think there's a lot of validity to the concerns you raise, but I would say that there are multiple ways of looking at this, and it's not just that this is a brutish Borg-like, we are, in the middle of Star Trek, we're taking away all autonomy and we're going to become some large autonomous machine. Far from it.
That's great. And there was a lot. And so I'm going to ask this kind of simple-minded question or two, which is I'm curious about what the adaptive part of it, what's this adaptive to? Because I'm thinking of the case where you have trials that are trying to, even if they're these kind of iterated adaptive trials for vaccine effectiveness, or vaccine efficacy, they may not be adapting to vaccine effectiveness, and which has very different criteria for what counts as success. And that maybe you don't want to, how many people, getting more and more people being effectively treated by a vaccine may also coincide, I can't think of a make-up example to make it, with fewer and fewer people being able to access a more effective treatment. And so you get this kind of inverse relationship between, if you bring the, so this is trying to talk about this gap between the clinical, which and the research, because the research is interested in what's the biochemistry, what's happening here, what are the effects in the body. And then when you bring it out to public health and say, okay, I got something really good, but how is it operating in this more complex social context, which has to do not with who would benefit, but who could benefit. Is it too expensive? Don't I want to get something that's robust against a lot of these different values that are being kind of satisfied in these domains? So I don't know if that was a coherent question, but I'm curious about what you're adapting to more successful biochemical responses to a medicine, right, or a treatment. And the other one, you're adapting to more or less access to some kind of treatment across populations. So you're maximizing different things.
So again, these are great but complex questions. So a couple of thoughts. So there's nothing inherent in a remap design that needs to just be biologic. In fact, a remap design is in many ways no different from what Amazon is using for testing your preferences on what ad you click. The underlying math doesn't mean that it's constrained to asking biologic questions. It turns out that so far, no one's really thinking about using these designs that much in policy, for example, although RCTs are being used in policy, sort of more traditional RCTs. And you can definitely think about applying it in policy if you could get policy agents to sign on for it. If you could get the Department of Health in Harrisburg to agree to take 40 counties in Pennsylvania and agree to some stepwise rolling random. I mean, it would be nice if they were progressive enough around that, and that would be great. Then you might still have these issues of these outcomes. And if you're adapting on a short-term outcome, but it's not by Prentice criteria actually predictive of the outcome you really care about. Even though I can answer that within the biologic context, the issue is still the same, which is, have any of these designs had any experience doing early adaptations on a short-term piece of information that correlates imperfectly with the long-term thing that we care about? And that actually came up very early on in breast cancer, because you want to run the trial over 18 months to 2 years. But whether breast cancer has been successfully treated is rarely judged over 18 months to 2 years. If you waited and followed for someone for 10 or 15 years, you wouldn't be generating evidence fast enough. And so there's a compelling need to try to see how the design does. Randomizing on making adaptations on short-term proxies. And so, for example, in the I-SPY 2 program in breast cancer, they've been validating the correlation between short-term outcomes and long-term outcomes. And they began with an intermediate outcome because they were too scared to use an even earlier one because it might not correlate well, even though they knew it would be great in terms of the speed. They thought it would be potentially horrible. And so they were looking at so-called, it's called PCR, pathological clinical response, or some aggregate biopsy score. And then they also had these imaging scores. And they have, over time, recognize that they've got an adequately robust correlation that they can now move more proximally. But that's well considered. And even in the simulation models, you can simulate to understand the operating characteristics of the trial, the consequences of making adaptations on an imperfectly performing short-term outcome.
So I have a question from the internet. It's not just random. So John Fuller, who's an assistant professor in HPS, is currently at NIH. He's been watching through the ether waves here. And he sends this question. He says, Derek, thanks for this great talk. I'm sorry I can't be there in person. I wanted to push you a bit on your claim that our goal in clinical research ought to be an accurate estimate for the individual treatment effect, ITE. As you point out, RCTs cannot give us a patient's ITE except when there's a low heterogeneity across ITEs in the population. However, I'm wondering how strong your objection to the average treatment effect is. Is your view that the ATE is not relevant to individual patient decision-making at all? Or can it still be useful in the absence of better information about the ITE? It seems to me that the ATE can still be relevant to the individual when the individual is uncertain about what their ITE is. In other words, who in the trial population they are most comparable to. Think about how we might reason when buying a scratch lottery ticket. Sure, before I buy the ticket, I would be great to know whether, if scratched, the individual ticket would be revealed to be a winner. Unfortunately, we can't scratch the ticket before we buy it. Yet, an expected utility maximizer can still reason about whether it would be worth buying the ticket, given information about the population of lottery tickets, size of the jackpot, number of winning tickets, cost of the ticket, etc. It seems to me that demanding that we know the ITE over the ATE is sort of like demanding we know what's under the surface of the individual unscratched lottery ticket before buying it. It says, I hope this is coherent. Sandy Mitchell will follow up and clarify on my behalf if it's not.
So, yeah, so, and I've been thinking about this. I knew I was going to have way too many slides. I had thought about putting up a slide that showed for a family of trials that all have on therapies that all have positive ATEs. When would I be happy just with the ATE? And I was thinking about on an axis of cost and an axis of potential for harm. Like, so if you put harm on the x-axis and cost on the y. So it's not like a cost effectiveness curve because every one is effective. They all have, but the plot looks similar. And then up in the top right, you have a therapy that is both expensive to give and some important proportion of patients have horrible side effects. And then there's another quadrant that it's really, really cheap and hardly anyone has any side effects. And then the others are. And then I thought, so first of all, I don't know what the distribution of all of these are, but all the ones where I think the drug is cheap, and so the cost to society of giving it indiscriminately is very low. And the cost to an individual is very low because, you know, even if it doesn't work. And so I really don't care about estimating more narrowly the treatment effect. All other three quadrants, even if I have an ATE is better than no ATE. But I would still rather be trying to get better conditional ATEs. And the very fact that no one has even tried when it is within our power to narrow it down seems problematic. And the reason they don't try is in part because trials cost so much money to do. And so they get their cohort and they limp to the finishing line, barely coming home. And that's just this habit. But if you really lowered the cost of doing RCTs, cost to patients and to the people doing the trials, so you could generate more, you could do larger ones where you could be looking across different groups, why would you not try to estimate conditional AT? So I'm not going to give up on the AT, but in 3 of the 4 quadrants, it seems to me it would be helpful to have Kates, because you don't want to indiscriminately give a drug that's really expensive, even if the patient wants it. Society ultimately is lifting the tab for healthcare delivery, and not including research, it's already 1/5 of the GDP. When you put in pharma, R&D, and all the rest of it, actually gets close to 25 cents on the dollar of everything we do is going into healthcare. And so I'm not actually sure that we should tolerate generating only ATEs for 3 of the 4 quadrants. Furthermore, that bottom quadrant is basically over-the-counter prescriptions. The other 3 quadrants are basically all the drugs we prescribe. So nearly all the drugs that are going to be physician prescribed, in my mind, meet criteria for trying to demand tighter ATEs, at least getting conditional average treatment effects. I'm going to finish with, so John leads with an analogy, so let me give him another one. ATEs are like having a radio in the 1950s. So without the radio, could you imagine? I mean, what a huge advance it is to hear music in your house from halfway across. On the other hand, think of the hiss, the background noise, it's coming through on mono, it's not a very precise signal. Imagine that in comparison to a high fidelity streaming Spotify that you could have today. So I'm not saying that you would give up on the radio, and in the 1940s, you'd rather have the radio than not have it. But I totally think we should be demanding high fidelity stereo. In other words, we should be demanding at least more robust Kates across plausible subgroups.
So this is a question that maybe just requires clarification. So let me go back to your analogy of the stadium. How there's no sense in rebuilding the stadium every time you want to play a game of football, because, well, presumably it's going to look pretty similar every time. But that's not quite the situation here, right? It's not actually the same game every time like it would be in a game of football. So I'm kind of curious about how these big studies which are simultaneously testing many, many different things with a weighting that is determined by the previous successes of those things, deals with new factors coming in partway through the study. What happens if somebody puts a new drug out and all of a sudden you need to take it in with the model that's already going to be biased towards assigning a different, or a treatment plan that's already going to be biased towards a different treatment? Presumably you had to deal with this in the COVID study, so I'm curious about how that went.
Yeah, so I don't know how recovery did it, because recovery wasn't using the domain structure that we use, but by having separate domains, if you launch another domain later, it has no effect on any of the other domains that are already going on. In the same way as, even if we weren't testing those other things, they would just be co-interventions that have been given non-randomly. And so it really is like a stadium in which you then just kept on adding on new games. And every time you added in a protocol, you added in a new domain, like someone shows up and, let me say this another way. You could add new interventions into the same domain. So you could have been testing antivirals, and then you'd thrown out 2 or 3, and now you've got a new one that looks best, and then someone comes in with a new one. You did A versus B versus C. You got rid of A, you got rid of B. C is looking awesome, and then someone arrives with D. And if everyone feels comfortable to randomize to D versus C, C is the new control arm. And it's true that in the trial, the head-to-head is the people who are being randomized contemporaneously. But we do have an overarching model that has other covariates predicting outcome like age, sex, all those things. Those things are estimated from the entire trial because the relationship between my age and my likelihood of outcome is more robustly estimated from thousands of me than from hundreds of me. And so the model uses all the information for the non-randomized covariates to estimate those, along with factors for time. But for the head-to-head randomization, that's still a head-to-head comparison within the trial. And so if you were giving C back in 2020, and now someone comes along and wants to test D against C in the year 2023, that's primarily a test of D versus C in 2023. Now, you can have as a secondary analysis an alternate estimate of the effect of C that includes C given back in 2021, but that's not normally trial-wise. But where it is like one stadium is when you bring that in, all you do is you file an amendment with the IRB. There's no brand new IRB review. The whole thing can happen in days. And all you do for the case report form for the data collection is you do some domain-specific data elements. But all the existing architecture for working out how you find patients, how you write down their age, how you load up, all the way the data flows, none of that needs to be restructured. That's all in place and still running.
More questions?
You said that when pandemic hit, before that your remat cap was already in place. I'm curious about how you used it during the pandemic, and also if there is another pandemic in the near future, how is this going to happen versus everyday daily care?
Yeah, so remat cap in particular decided to focus on what we call community-acquired pneumonia, both because community-acquired pneumonia is still a huge worldwide problem in non-pandemic times, and because most pandemics are community-acquired respiratory infections. Not all of them, but most of them are. And so, and because some of the treatments are not targeting the pathogen, but targeting all the support structure, like supporting the host, supporting organ failure, like respiratory failure. And so some questions, you might actually want to borrow estimates where now, like, what's the best way to ventilate a critically ill patient with pneumonia? It might be that the treatment effect only modulates, they're like two different subgroups of the same question, whether it's an influenza pneumonia or a COVID pneumonia. And so there was a lot of synchrony within that clustering. And so this same trial design is also enrolling in testing patients in flu and in bacterial pneumonia. And actually, there was an email discussion today about if H5N1, would H5N1 be a separate stratum from regular flu, or would it be all inside flu? So right now, it's still prospectively all inside flu. If the WHO declares it a pandemic, will we flip into a pandemic mode specifically for H5N1? And actually, we were in contact with colleagues at Imperial College in London today, just about the discussion of the likelihood that will actually become pandemic, which will be relieved to know they still think it's low, because it's still not doing proper air transmission between humans at this point, which is true for all the H5s. But the model is capable to go back into pandemic mode for other ones. And but if it turns out it was like a pandemic that wasn't an airborne virus that was predominantly pneumonia, then I'm not even sure remat cap makes entire sense. There might have to be an entirely separate structure. I don't think we're going to have pandemic Shigella infection or malaria, but if it was, it would be a different platform.
So it seems like remap is very much in the game of interventions that are already out there. I'm wondering if there's any lessons here for pre-market trials, phase 2, phase 3 contexts, or if that's kind of not part of the game, since that's more of the pure research program that's passionate.
Yeah, so I'm quite nerdy around the methods, and so I talked about RemapCap because it's actually been deployed in thousands of patients, but most of the methods are agnostic to the setting. And indeed, a lot of the original theory around adaptive. So Remap, in comparison to the I-SPY2 program in breast cancer, has these multiple domains going on at the same time. Whereas I-SPY2, which is also an adaptive platform trial, doesn't really do these multiple domains simultaneously. But I-SPY2, which got up and running many years before Remap, and they're sort of inspired with similar by each other, with similar statisticians working in both, it specifically was in the pre-registration space. And what it was trying to do was tackle the problem of transitioning from phase 2 to phase 3. And that's a relatively inefficient space in trial discovery. So people don't have good endpoints from phase 2 trials. And so they tend to do they're like underfunded phase 3s, where they then go on a hope and a bet and a prayer around what they should take into phase 3. So this I-SPY2 is actually trying to test multiple candidate molecules that have emerged out of phase 1 on short-term outcome measures to then be predictive. The outcome is actually the likelihood that if tested in a phase 3 trial, they would be successful. And the whole thing is about trying to do a more efficient selection process for what goes from phase 2 into phase 3. So definitely pre-approval, these designs can also work. Another issue here is whether the FDA would accept these designs. And I had one line on regulatory oversight. RemapCap has actually included full-blown registration trial elements in it. So we were testing some drugs for Amgen in the RemapCap, where we were doing one-to-one randomization with placebo. It was on the architecture of all of the data collection was in the Remap. And then we had an extra module for the extra data that the FDA wanted collected because this was an investigational drug that wasn't approved. But again, that's better than starting from scratch. You go into an existing trial, you get all of the staff hired, you've already got 80% of the data collected, and you add a module for the extra data. And the trial was able to it can still set that there'll be fixed randomization within one of the domains while all the others are randomizing. Now, we would have rather done response adaptive randomization, and we have also run combined phase 2, phase 3 trials that are fully Bayesian with response adaptive randomization fully approved by the FDA. Including, we ran one that would have even switched from phase 2 to phase 3 during the trial, and we wouldn't have known. Like the rule would have pitched it. Turned out the drug didn't work, but the point is that the FDA totally signed off on it.
Yeah, this is sort of a naive question, but I'm wondering how the choice of variables for the conditional treatment effect works. I mean, it's just the sense of the researcher that some of these are more likely to be important or consequential than others. And relatedly, if you get disparate treatment effects according to type, condition on these variables, is there some way of kind of following up with other studies where you can see whether the presumed conditional treatment effect is actually, somewhat stable across studies? Is this something people worry about, or is the answer just obvious?
Oh boy, do we worry about that. And so this would be a classic example of part of my drive-by in this talk. So these are fantastic questions. So Kate's. So part of the reason why many people have defended only producing 8s is that no one trusts any Kate's. So for many years, frequentist statisticians would have said the best estimate for a subgroup is the overall trial. In fact, an Oxford statistician, Stuart Pocock, famously wrote that the best estimate for any subgroup of the trial is the overall estimate from the trial. And they're basically saying that,
But you do specify the relevant variables
well,
ahead of time, don't you? You don't look after the outcome.
hold on a second. I'm going to get there. I'm going to get there. So there is a prevailing bias that you shouldn't even have tried to do this. Although then lots of people have realized in retrospect that was a mistake. So many of the big cardiology trials out of the Oxford Clin-Epi, so Richard Peto was not a fan of doing any subgroups. And then a lot of people in retrospect have realized that many patients were inappropriately treated for many years afterwards, and only later they took, like, what were we thinking? It made no sense that these patients would benefit, etc. When you go to generate K, the way people did it traditionally for years was they would just show a long table of subgroups. Men versus women, old versus young, those with heart disease versus those without, or those with underlying COPD, etc. That's very crude. Those are chosen based on biologic rationale. We believe plausibly it might happen, but they all have wide confidence intervals around them. And the interesting thing is that when you do subgroups, that's just one variable at a time. So for example, men and women in a trial are different in that they're men and women, but they're more alike than they're different. They have the same distribution of severity of disease. They have the same distribution of the likelihood of having underlying comorbidities. And so they actually have a hard job being very far away from each other. Even if the distance is 0.5 odds ratio, if you plot that out on a typical distribution, that's still pretty close to the median. And so traditional subgroup analyses are actually kind of awful, but they were the predominant mode. And then they were tested using something called the Breslow-Day test, which is famously underpowered and nearly always returns a value of we could not find evidence of heterogeneity. So that permeated the literature for decades. There's two big movements in trying to estimate heterogeneity of treatment effect these days. One is modeling based on risk of outcome. So that's a supervised learning on the outcome, but with no knowledge of the effect. And you can build a multivariable model. You can pull one off the shelf from a particular disease, or you can build it just on the control group and then estimate the treatment group, or you can build it, and you can do split house validation, or you can decide it's all right. You can do anything you want. You could also build a direct effect-based model, where you include a covariance, you include modeling for so you have it's supervised on the effect. So the model actually knows. So that would hopelessly overfit. So you can't run that in the whole trial. So that you would need to do some sort of split house validation, and now the preferred model in that is to do like a generalized random forests to try to protect, so like a machine learning model to protect against overfitting, but also still only in a subsample. And actually, a lot of that work isn't happening in medicine, it's happening outside medicine. So it's folks like Susan Athey and Guido Imbens at Stanford, or actually, I was talking about this at lunch, Josh Angus, who got the Nobel Prize in economics for his work in this space, who, by the way, is from Squirrel Hill, something I don't know why the city doesn't celebrate it. And those models are very interesting. They're giving a direct mathematical estimation of the effect. But the price they pay for that is that it was totally supervised. And so then the huge threat is the generalizability, which is why you have to think about how you extend it. And so then even if you do split halves, then there's question of what's the best way to get split halves in the trial. So we're running one right now where we're not only doing split halves by patient, but by the site they were at. So that, because there's still a little bit of independence between patients if they were treated in the same site. So we're trying to build the effect-based model in patients who were treated at sites that were never completely separate from the patients and the sites to try to keep them as far apart as possible. And then still you would want to then validate, for example, in other trials. But when I say validate, you still have this Paul Holland problem of counterfactuals. The only place that I know of where you can really do a head-to-head on the different modeling projects is totally in simulation, where you can actually create spiked data sets and have forced interaction terms with treatment, etc. that then have to be discovered by different modeling approaches to try and assess their performance. But it's a great question. So.
Yeah, so I know we talked a little bit earlier about this, and I know the focus here is mostly on intervention, but I am wondering if you might be able to draw some analogies to diagnostic reasoning. So we've been talking about like making decisions in the face of uncertainty, and I was really interested in how you alluded to automation in the EHR would like, reduce that sort of cognitive load in physician. And I'm interested if you have the same sort of intuition about diagnosis as you do as treatment.
Right. So we actually ran this thing for the National Academy last year on diagnostic excellence. It's an initiative that's been begun by the Gordon and Betsy Moore Foundation. And so they've been funding a bunch of us to get in a room and think of what does diagnostic excellence mean? And I came to this group with a huge degree of skepticism because I thought, what is the value of a diagnosis other than the consequence of having the diagnosis? And there's a little bit of, well, sometimes it's nice to just relieve certainty, you know, it's now not something else, etc. But most of it is tied to the decisions that you then make afterwards, including even prognostic significance, which has implications for stop treating and just go to palliation. And so I felt like it's clinicians who get enamored with being the master diagnostician. And I felt it was totally overrated because what matters is what you then do afterwards. And so in that way, most of diagnostic excellence is just parsed out by, you could still follow it through to the decisions made. And indeed, we have, for example, in the space of pneumonia, biomarkers that people say, this will predict whether the patient has pneumonia. But people have debates around what is pneumonia. And so you can do an end run on that by saying, see whether a biomarker sharpens the ways in which you would then make antibiotic decisions to then result in a maximized outcome of patients getting well with the fewest antibiotics used, and skip past whether they ever actually, like whether they had pneumonia or not is less relevant than whether as a cohort, everyone who might have had pneumonia gets the most parsimonious path in terms of least antibiotic use with sort of most people surviving and no antibiotic overuse. So part of me, when you say you're interested in diagnosis, I would say, great, just tie that to treatment. But if you want to sort of stop and play with just diagnosis, I still think it's also true that, and it also gets to the question that was raised earlier, that I'm very interested in all of the choice architecture at the moment that a physician is thinking about something. And I'd actually like to deconstruct some of their rules of thumb and all they're here, because sometimes they're certain where they have no right to be certain. Like they're certain because it's the way they've always done it, which is not the same thing as they're certain because they know the answer. They just think they do. And the EHR definitely provides a wonderful option in theory for providing all sorts of, you can play with defaults, you can play with framing, you can play with choice of diagnostic tests, you can play with better organization of the information to get a more accurate pre-test probability. You could actually feed back to the doctor, if you order this test, given these here's how much more certain you could be. There's a ton of things you could do. And since you don't know which one of these things would be best, you could actually randomly do different things. Like you could randomly give no information and just let them go on hunch, or you could have more than one way of providing the information and see what goes over best. And I think it's an almost unlimited choice set.
Any further questions? All right, before we thank you, we have a parting gift that every speaker at the center gets. It's this umbrella, which we don't, I know it's a sunny day, but give it a minute. Thank you.
very much.
Welcome to join us for our reception out in the lobby.